Let's record.
I have a quick
Okay,
question.
perfect. So recording is live. All right. So the last thing I want to cover. Okay, so this is an interesting question from Guy, I believe your first name, Mats. AI is the way to go. How do you feel about quantum? Very interesting quantum computing. I don't know if we have time to introduce it this semester. It's more like my time to read up on it so I can discuss it with you guys. But definitely I want to introduce it in the class, if not this semester, later semesters. There's actually one faculty from, I think it's Queens College at CUNY, Norson Janowski is the last name, from computer science. He has a book on quantum computing. Of course, there's many books out there, but if you're interested, you should definitely read up on it. The Hunter College Library, if you search there, quantum computing, they have a lot of ebooks available for free. That's always my source for materials. And I've spotted a good couple of books, guys. So if you want, I can point you to them to read. Yeah, hopefully we'll introduce it, if not in this course this semester, like subsequent semesters. Okay, so if there's no good questions, so if there's no other ones, let me, before I go to the course material, let me say the last sort of operational kind of thing. So how are we going to be doing our homeworks? So this is the Google Collaboratory, which I have linked here on the Blackboard, collab.research.google.com. So that's where you're going to be doing your code. And then I'll explain how this works. Some of you might be familiar already, just working on it with it on your own, or haven't taken my previous courses. But for everyone else, do not worry, I will explain it. So this is an online interactive coding environment for Python. It's free, and you just need a Google account, which you might have already, or you can make a new one to keep things separate from your personal data. I mean, Google, the accounts are free. And that's where we're going to be doing our homeworks, or if you want an interactive code environment to practice the Python. And you'll see today as I go through the lecture, I'll be pasting the codes in there from the lecture to demonstrate them. And then the homeworks, you can actually do it. You can actually, you'll do it in the Google Collaboratory because that's where it's required. But you can actually share with me as a response to the homework your Google Collaboratory, or just take a PDF, a printout PDF of your code, and send that as a response to the homework. With the sharing, because sometimes like you have all these options like share with access, with email login, and you have to specify the email. And there's the other option which says share with everyone. I think that's the preferred one to make sure that I have access. But again, my experience is like always something goes like off with the sharing, and then I try to correct the homeworks, and then I cannot access. So I think it would be preferable just to do a PDF printout and send to me, because it's tedious to have to go back and forth and contact people and say like, yeah, you send me the link from the Collaboratory, and then I cannot, where you have your homework, and I cannot access it. So they try again, maybe then it works, maybe it doesn't. So I think PDF printout is better. And when I say share in the Collaboratory, it's like part of the Google Drive, and then it's shared like any other Google Drive document, with a link. Now when I say, let's start a new notebook actually, and when I say send me the PDF or share in the output of the homework, I mean not just the homework to be considered, and I'll note that when I post the homework as well, but I'm telling you now, is going to be considered like a full answer when you have your code and you have run your code, and you'll see as we run the code here, it generates outputs, and the outputs, the code and the outputs are there without any errors. So that would be considered the correct answer to the homework. So you run the codes there, it produces the output with no errors. Obviously, for your code in the homework to be correct, it has to have no errors. And then you take a PDF print out of that and you attach it, or if you want to do the link with the sharing, that's fine too. But just hopefully, won't have to go through any troubles because the link sharing doesn't work. Okay, so the Google Collaboratory, really quickly, for those of you who don't know it, again, it's like a Google document. It's you'll see here is a IPyNB interactive Python notebook. It's an open source project. It used to be from the interactive Python, the IPy before, but then it became Jupyter, which is where the Jupyter book comes from, but it's another branch of the project. But this is basically the Jupyter Notebook open source project, which Google took and they integrated on the Google platform. There's also the you can try it from Jupyter Notebook from your browser on the Binder. I'm fine if you want to use that. For those of you who are familiar, if you're, say, for example, you're a fully open source advocate like I am, and you're anti-Google, whatever, that's fine for me too if the code works. And you might even have your own Jupyter Notebook installed on your laptop. That's fine as well. But I use the Google. I'm anti-Google too, but I use the Google because it's the most easy for everyone. Now, for those of you, if you're planning to use the your laptop or the I haven't worked with this one. Some of the libraries we'll be installing when we'll be getting into like the machine learning stuff, they're pretty big, and on the Google they work just fine. So I'm just saying on your laptop, you might be installing those coded libraries and it might be eating 1 gigabyte. So just a warning there. It's more convenient in that sense to use the Google and save the space. Okay, so we said this is an interactive Python notebook, and like any other document on the Google Docs, you can name it as you want. So we can name that lecture 1, IPyNB. Let me go back to the Zoom. If you have any questions, no questions, okay. So you can see here upload, open notebook, save a copy in Drive, so on and so forth. So if you work, say, for outside on Jupyter, you could actually upload it there and continue the work on Google. For the files, we'll go over it again, but I can show you now. In the first lectures, we won't need to import any files, or we get them like through a remote fetch command over the cloud. But here's the files icon here. So see, when I clicked actually the files icon, it connected automatically here, and you see now that you have how much available RAM and disk you have that Google gives you for free. But if I haven't clicked the files, you could also click here to connect and get that. So here's the files application. You can upload files, and I'm referring to that when we have some files that we need to load the data and parse them for the homeworks. Now this, you can make folders, upload wherever you want, and then on your code, you see where it says you can click this one after you upload it. Let's say we can go in here, and let's say we uploaded this. This is machine learning data actually. which Google has preloaded for whatever reason. And let's say that was the file we uploaded, through the upload button here. We put in this directory, and then when you use an R code, you say copy path, right? I'm just showing it now because people, it's always hidden there, cannot find it. But that's the most important thing. You want copy path, and then it's a little / content sample data, but this is the path you refer to in your code, your Python code to load the file, right? So just remember that, but we can go over it again, right? And then after you've written your code, you can do a print, like I said, and generate a PDF, and that's what you send as a response to the homework. And I think that's basically it. Here, I'm not going to use it, but for those of you who know Markdown, you can actually write Markdown. So it has like, well, let me say, so what you see here, this is called a cell, right? So this is a code cell where you write your code, and you can create like more cells with the code, but also you can create text cells as well. And the text cells, I guess, as you see here, you can write like regular text. So again, that's an interactive notebook, so you can mix and match like text where you describe what the code is doing, but and the actual code which you executed, right? And you could consider that being also a live executable book as well, even though it's much simpler format than the Jupyter book I'm using. And then here you can write as regular text, and you can write as Markdown, right? So it's helpful if you want to write the whole homework, the questions part, the theoretic questions part, inside here with a text. But if you want to write the coding part only here, and then the questions part in a Word document, that's fine too, whatever you prefer. If you want to put comments in your code just to make it even more enhanced, that's welcome as well. So you can use these text options of the codes here, and then you'll see now how we write the as the lecture goes along, how we write the code and execute it. And I'll leave it at that. And let me go back to the Zoom. I don't see any questions on the chat. Is there any questions from what you've seen so far?
Yes, I have a question. Can you hear me?
In the collaboratory? No, okay. So I guess we can go ahead with the lecture. Of course, again, we have the question. There's a question by a guy. You're typing it. I'll wait for a little bit if you want to type it. You can try also speaking the question, but my sound is not working, unfortunately. Yeah. Is there a good way to clean up the code as you go in? Okay, that's a good question. If you mean like in the sense if it creates errors, it's very simple. Basically, you just rerun. So if I write some code in that cell and I have an error, I can grab one right now so you can see. I'll grab it from here. Let me, and I'm just gonna introduce an error on purpose. So let me paste that code here, and then I'm just gonna remove one of the parentheses so the error's out, right? So you see the error. So basically, like you just do clear output, right? And then you correct the error, and then you just, oops, I lost. Okay, there we are. Just lost my screen suddenly. Okay, and then correct the error, rerun it, no error, and we're totally fine, right? So that's how you do it. Yes, it's good to, it's totally fine to write in your total IDE. Like I said, some of the libraries, like for example, when we do like TensorFlow, this is like almost 1 GB library, so it's going to be in a lot of space from your Colab. I mean, you can even paste the code output from your IDE, that's fine. That's fine with me. And then we have a question: will this course be difficult for students never coded? I think it's actually going to be very difficult. Even the first lecture will be very difficult because this course, the prerequisites, is programming. Again, if you had, say, some programming background in like Perl or whatever, and you didn't know Python, I mean, if you learn one programming language, you can learn others pretty quickly as long as they're similar. Perl and Python is similar. So you knew Perl, you could pick up Python with very intensive study the first 3 weeks and probably be able to continue the course, but never done programming before. I think the course is going to be very difficult. So my suggestion would be, because I don't want anybody to fail in the course, probably look for an alternative. Yeah. So that's my advice in regards to that. Okay, so if there's no other questions, and for the interest of time, let's go ahead with the lecture, and then we're gonna keep going back with the Google Colab. Okay, so that's the slides here. So the topic of the first lecture, and because I'm actually going to be going back and forth every 5 minutes, or maybe let's say every 2 slides between the Zoom window and the slides window. So if I don't answer your question immediately, then when I'm back, I'll see it on the chat because I cannot hear the sound now, and then I'll answer it. But we'll fix the sound by the next time. Okay, so the topic of the first lecture is graph theory and network analysis in bioinformatics. And then I have 2 links for just general information for the Wikipedia definition of graph theory. And there's an online course from the European Bioinformatics Institute where I borrowed some of their images and their Creative Commons license, so I can reuse them with attribution. And that's how I make my book too. Creative Commons, and others can reuse it with attribution for non-commercial purposes. This one, as I said before, right, everything that's linked, it's also part of the reading material. So you definitely have to read that stuff. Most of the concepts we describe in the lecture anyway today, but for answering the homework, you might have to go back and read. And the Wikipedia article, I think it's fine. It's more for your extended interest, if you will, if you want to read it, right? But definitely this one for the homework. Okay, so we'll discuss basic graph theory and network. You can call it interchangeably graphs or networks. Concepts and data structures for those, which are central data structures for bioinformatics data. And again, like graphs or otherwise networks, however you want to call them, are one of the most common ways to represent bioinformatics data. And what are some examples of those data? Interactions between genes and proteins, genetic regulation, one gene regulates the other, or a protein regulates the gene, can be represented as a graph. And we'll see how a graph or network, however you want to call it, looks. And then also interesting, like molecular structures, protein structures can also be represented as graphs. Essentially, because you'll see the graphs are kind of balls and stick structures, like sticks connecting balls. So if you were to represent a protein as the protein amino acids as the individual balls, and the links between the amino acids as those sticks that you see the graphs, the protein structure can be easily modeled as a graph. And you would ask, what's the reason to represent a protein structure as a graph? The reason is more general, how you can represent real world data in a computational data structure that you can apply algorithms on it, right? So if you want to do protein structure prediction, for example, which we're not covering so much in this class, maybe we'll touch upon it a little bit in the when it comes more to the machine learning stuff, because there's huge applications of machine learning to protein structure prediction. Then we'll do it there, but not going to be one of the core topics of this course. But again, the take-home message is you need to represent real-world data and some data structures. In this case here is the graphs, which you can compute upon this data, right? And beyond the molecular biology, the graphs, it's a very generic data structure. For example, if we were doing ecology, we could represent species interactions. Beyond the gene interactions, we could represent species interactions with the graphs. And now the big question, what is a graph? Essentially, it's a mathematical structure or a data structure when it comes to computer science, capturing binary relationships, meaning like relations between one object to another object. And those objects called nodes or vertices is a more mathematical term. Nodes is a more computational term, or I said very plainly before, as you'll see in the next pictures, the balls, right? Little, because that's how we draw them. I mean, you can use squares if you want. That's totally fine. And then the two objects, the binary have the binary relationship. They are linked together. So that's the stick between the balls. And the official term of those links is edges, if you are a computer scientist, or vertices if you are a mathematician, right? So balls linked together by sticks, and mathematicians call those balls nodes. Sorry, the computer scientists call those nodes, and the mathematicians call them vertices. And then the links between those are called edges, right? But we will use the nodes and edges terminology here because it's a computational course, right? Now the edges, the links between the objects can be directed or undirected, meaning having an arrow, and weighted. You can think of weighted or not. Weighted meaning a thicker line or not. And then also each of those objects, whether it's the nodes, the balls, or the edges, the links, they have attributes. They can have attributes attached to them. Okay, so let me see. Well, leave that for now. I can try to explain it here with the text. So when I say directed or undirected, meaning that say we have a relationship between two genes, gene A and gene B, right? And if you have a directed edge, a directed link, meaning like an arrow going from gene A to gene B, that will be representing a directed interaction, meaning that gene A controls gene B, right? So again, this is made as such so that you represent real world data, right? This mathematical and data structure is used to represent real world data. So if you want to model gene A regulating gene B or protein A regulating gene A, then we put a directed edge going from protein A to gene A, right? Or undirected, meaning that either way, they're mutually regulated or whatever. And weighted or not, essentially that's the attributes that I say on the next point. It would be that this link, it would have a certain weight into it, meaning how strong the regulation is or how strong the interaction is, right? And both the links and the balls, the nodes, can have attribute attached to them. And what would be one attribute? The name, for example, right? Which gene is represented by that node. And we'll see that in the next slides with the code. And let me go back to the Zoom window for the questions. And there's a question: can links have signs, like for up, down, or down regulation? Yeah, so that exactly what I said about the attribute attached to them. So that would be an attribute of plus or minus, which would be on the link on the edge, representing up regulation or down regulation, right? So you would code that for the property, and that's a whole. And you'll see that as we move along in the class and the homework. So that's the whole idea, and not idea, like research and challenge, like how you can set up the data structure. I mean, the graphs is a very generic data structure, and then it's about like how you utilize these options that it has for directed, undirected, weighted, or not weighted properties on the nodes and the edges, so that you can model. That's a lot of the art in it. So you can model the real world interactions and knowledge on the data structure as good as possible. And then, of course, once you have, that's the first step, once you have the data in that data structure, then you need to write the algorithms to process the data and get inferences and discoveries from the data, right? And of course, it's always us as researchers and code developers to code the data correctly, right? So as I said before, the plus and minuses for the up regulation, down regulation, you would put it as properties on the links. And here's an example of a real biological graph. You can follow the link here on the paper if you want to read more details. I just use it as a schematic here for demonstration. So you can see that what they did is that they have directed edges here. As I mentioned, like this can be, for example, a protein that regulates these 4 genes. I guess here the color doesn't matter. They just highlighted probably for the research in the paper some gene and its interactions in this network. And then, as you'll see, we'll do today in the code, they put some names on the nodes, and I guess I haven't figured out how to do that with network X. I'm sure it's there already. That's more like a visual way to code attributes. I'm sure it's an option in network X. Like some nodes, they're rhombus shapes, some they're circles, some they're rectangles, and so on and so forth. I mean, it doesn't really matter. Again, that's a visual thing. As you'll see when we'll be coding today in the examples, like whether it's a gene or it's a protein, we just code as an attribute of the object in the data structure. And of course, then if that's an option in network X, which I'm sure it is, then you can write in your code, if the attribute is a protein, draw it as a circle. If the attribute is a gene, draw it as a rectangle or something. If the attribute is a chemical molecule, draw it as a rhombus, right? Whatever, however you want to code it and represent it, right? But again, the shapes here is more like a visual thing. The key is the attributes and the data structure, right? And from what I'm seeing here, the data structure is a directed graph. Okay, and then let's go to the next one. Okay, so protein interaction network graphs, a very common thing you will find for this type of data structures. Essentially represent physical relationships among proteins present in the cell. Just to give you an example, if you recall your BIOL 200s or 300s or any other biology course, for example, when you want to have transcription or transcription being DNA becoming messenger RNA, and translation being messenger RNA becoming a protein. you have a whole conglomerate of proteins coming there together, 5 proteins that make a complex, and that complex is the DNA transcription complex, which generates mRNA from during the transcription process from a DNA template. And those 5 proteins, 5, I'm just making a number, they just come together to form that complex. They form an interaction network, right? Or another interaction network, you might be in the metabolic pathway where you have like 10 different proteins interacting at different stages together, so they can convert, say, complex carbohydrates, starches, to like plain sugars, which is what happens in the metabolism during the cell, right? So those 10 interacting proteins in that metabolic network, you could represent it as a protein-protein interaction network, and so on and so forth, like signaling networks. But this is biology. We just explain it quickly here. Our focus again is the programming. So in the protein-protein interaction networks, you would represent the protein as nodes, right? And their interactions as edges. Undirected, they can be directed too. Okay, and then those, why do we mention it here? Because proteins obviously is essential to the cellular processes, and protein-protein interactions, as I mentioned, the example with the metabolism or the transcription, the translation, are the most essential cellular processes, and they're very well studied networks in biology. And in fact, we have a lot of data out there, rich data sets we can use to and we will use in this course to learn and practice our code, but we can also use those bioinformatics data that are out there to do research and publish papers. And that's a lot of what we do in bioinformatics, right? We get data sets from the databases out there, and we write algorithms to analyze those data, and that's the research we do. And then some of the algorithms, for example, is finding in those protein-protein interaction networks some proteins which are very highly connected. There's graph algorithms for that, and those are the proteins which are essential for the survival of the cell, right? And if those proteins misfunction, they get mutated, for example, you have like bad diseases like cancer or other diseases, right? One you might know, you've taken courses before, you might have heard the p53 is a central protein which interacts with a lot in the cell, and if it gets mutated and those interactions break down, then you have cancerous cells, right? And then Wikipedia article, again, when I post Wikipedia stuff, it's more like for your general interest, right? You don't need to read it. The stuff you need to read is the code in libraries. And earlier today in the lecture, when I had the link from the European Bioinformatics Institute for the network analysis in biology. And then I have a question, which is there a graph for mRNA and protein interaction for SARS-CoV? Yes, definitely is. I can remind me that in a discussion forum. And if you're interested to learn more, I can post a link for a public data set which exists for that, so you can have a look further. That would be actually a good research project for somebody who comes out from this class and really wants to spend time to do research on these topics, because there's a lot of data obviously out there now with the COVID interaction data. Okay, so let's see if there's any other questions before I go to the coding. Okay, no questions. All right, so what time is it now? It's 11. Maybe before we go to the coding, we should take a quick break for everyone to rest. So let's take a break until 11:10. And actually, since everyone is here, let me take attendance now. So let's switch to the gallery view. So you can turn, let's take attendance. So you can turn your screen off if you want, or for whatever reason, if you don't want to turn it on, just let me know in the chat. Okay. Okay, so also for those of you who write on the chat, it's actually on the chat, sometimes I only see your first name, so it's better to say, some of them I see your first and last name, but better when you type on the chat, just say like your full name and say I'm present, right? So, okay, so attendance is taken, perfect. All right, so let's do a break for until, let's say 10 minutes, 11:15, and then we'll be back to continue after that. So you can turn off your cameras, we're taking the attendance, and then we'll be back at 11:15 to continue with the coding. And think of any questions you might have during the short break so we can answer them right after. So see you at 11:15. Okay, so I hope everyone is back from the break. If there's any problems hearing me, please let me know, just to make sure I'm not talking muted. So good, you can see my screen. Okay, perfect. So let's continue back to our coding, and let's use the Google Collaborator as well. So let's create a graph in Python. All right, so two important things. You always need to install the libraries. So here, I guess I could have put it in that little box here, but either way is fine. I just wanted to put it here to emphasize. So what does this pip install command do? Essentially, it installs the programming libraries, right? And it's a little, let me put it on the Google Colab. So going to open, so you see here, actually, we have open a code cell below or a text cell below, or open a code cell above, right? So we need to put it always on top. So why this exclamation mark? That means like execute one command in the what it's called the terminal, which we're never going to use it, because essentially what those are on Google Colab is a small virtual machine, a virtual computer that you get with, as you see here, 0. Less than 1 gigabyte of RAM, small machine, and how much is it? 12 gigabytes of disk or 41 gigabytes of disk. But then it's Linux virtual machines, and what you do is installing the libraries we need, the matplotlib, which is going to be used for drawer graphs, and the network X, obviously, which is going to be used for our networks. Now let's copy that there and execute it so you see how it works. And the thing is that just to always remember that if you go away from your computer, this actually, or close your computer or turn off whatever, this is actually going to disconnect, and then this gets wiped out, if you will. Essentially, the virtual machine shuts down. So when you reconnect, you get a new virtual machine, and those, you might still see the codes there, but then after you hit connect, you have to execute it again. Essentially, if you go away or close your computer, you have to execute all the cells again on the Google Collaborator, right? So that's to remember, because I always get the question, is like, oh, I came back and then I've worked through the 5 first cells, and now I'm clicking the 6 and I'm getting errors. Yes, because you have to first execute the very first one to reinstall the libraries, because you get a new virtual machine, a new computer on the Google Cloud. And also, like, if here you're creating variables which are executed, are used later on in the code, then you have, because you got a new virtual machine, those variables are gone, so you have to run all the cells again, right? So that's one very thing to remember. Always, when you go away and come back, just rerun it from the beginning. So let's install those libraries, right? And it's very easy. Essentially, it shows you the terminal output here. Some of you, maybe you've taken courses or recently where we use those. I don't think anybody besides the bioinformatics use them. Maybe in computer science, where you use like servers which are in the Hunter, and you have to log in through the terminal and stuff. You can see this one is abstracts a lot of that stuff, right? Okay, so this is all installed now, and then after we installed it on our quote-unquote terminal, now we're going to work the Python and import those libraries, the network X with as short as NX, so we can use it, and then the matplotlib. From the matplotlib, the big library matplotlib, install the Python plot, which is what we need. Okay, so here, let me actually enlarge so you can see a little better. So here I'm going to create a new instance of a graph. And this is an undirected graph. We'll see later option for, for a directed graph, and also you can see it in the library. And then we're just going to add a node into that. We could name the node, we'll just put like an integer 1. We can name the node whatever we want. I could put a string in there for the node name. But you see here one method to add a node. After initiate my graph, add node 1. But I also can add nodes more than 1 at the same time, but just using a Python array or list, if you want to use the more official term for Python list, with nodes 2 and 3. Again, that's just integers, right, that I'm using. I can name that node if I wanted hello, right? But I want to keep it clean, and my node, because the object I'm creating for the node is just represented by the integer tool, right? So let's run that. As long as you don't get any error, it means this data structure have been created and it's in the memory of that virtual machine, right? So now we have a graph with 3 nodes, right? And I'm going to open a new code cell, and then I'm going to go back to the slides and run the main code. Let me check on the chat. There's a question. We will have to install this in every session. Yeah, that is correct. You're not going to have to rewrite it, because this Google Colab that I created is on my drive. So if I close my computer, I close this tab here, and then I go to my drive, Google Drive. I'll see that file. I click on it, and then it takes me to that, and all this stuff except the output, of course, it's going to be there. So I just need to reconnect and rerun all of them from the beginning. And I think, yeah, so you can do run all, just to save you clicking on each one individually, right? Okay, so we created the first one, network X reference, again, undirected graph, as I said. And then we only now have a graph. We just created like 3 nodes, right? 3 balls just sitting there in the air, 1, 2, 3. But we need to connect them with edges. So let me get that code. And I'm trying actually to find how to add a link there. So instead of copy paste, I can just click and import that code automatically on the Google Colaboratory. I might have that the next time, but now it's copy and paste. Okay, so I added this code here. So what I'm doing is I'm using another method. Again, you go to the network X and read it, and then you see the add edge method. It's connecting, adding a link between nodes 1 and 2, and I can put an attribute of this edge to be color red, and weight 2. Now these colors, it's pretty straightforward to figure out. It's like G is green, B is blue, and so on and so forth. That's how network X represents it, and the weight is just an integer, right? Now you see here, like straightforward. I said add an edge between 1 and 2. There's another way to add edges, as we added like a bunch of nodes at the same time. We use what we call the Python tuples, which is kind of like the list we've seen before, but this with parentheses. Tuples, for those of you familiar with Python, is lists which are immutable. You cannot change their data. And then we just specify the tuple, and then we just say add edge, call the method add edge with a tuple. One other thing I want to show, the nice thing with the Google Colaboratory, I'm not sure if the Jupyter Online has that. Some of the IDEs you use locally, they might have that. When you go over a method after it has imported the network X library or whichever programming library, it imports also the documentation of that library. So when you go over the method, it can give you a quick overview what that method is doing, so you don't have to go back and forth between a window with a code and a window with a library to read it up. So you can quickly see the examples how to write your method or your function correctly. And I think open a new tab, yeah, so you can see it on the side too, so you can refer to it, right? So that's a functionality you can leverage. And also another thing I want to show you, let me type it here. If you do like, so the G is a graph object, right? So if you put dot, then you see the list comes down, which shows you all the methods that are in the G object, which is a graph. When I say object, don't confuse it with the object when you say graph object. When I say object, I mean object-oriented programming for Python, right? And then see, I selected the add edge method, and then when I go over that, it can give me the documentation for that method, because every Python object has a set of methods, and every method has its own method or function is the same term, has its own documentation, right? And this method obviously is for adding edges, links between the nodes in the graph, and here's the documentation, right? So I can close that and clean it up a little. So remove that code. Okay, so we see how to add the edges. I added one between node 1 and 2 with one approach, and nodes 2 and 3 with another approach. And then I'm going to go add 2 more nodes, 4 and 5. So we have 1, 2, 3, 4, and 5 nodes in our graph. And then that's another way here to add edges. Basically, you specify add edges from this array, which contains edges between a tuple. You saw the tuples before, so now you put like 2 tuples at the same time. So an edge between 3 and 4, first tuple, and an edge between 3 and 5, second tuple, right? So now we added the edges. We added additional nodes. We added edges between those additional nodes and our existing ones. And now we're going to draw the graph. So NX, which is a short for network X, we call the method draw, right? And then again, if you, the NX is a network X library, so you could go here, and then it just gives you like all the methods of the library, right? So I use the NX draw. So what do we want to draw? The graph G, which we created, and we want to have labels in this graph. That's why we say with labels true. And our font weight, we want it to be bold, right? And again, go read the network X library to see what parameters you have to customize that. So let's draw it. And here's our graph. Pretty simple, right? You can follow the code to see the links that we created. If I redraw it, probably it's going to have a different shape. Yeah, because I'm not using, so see, I'm rerunning the cell and it's redrawing. So it has a different shape because if you read the network X documentation, you'll see like you can specify the layout of the graph. Layout meaning like how to position the nodes. Here, I'm not specifying it, so it just picks up a random one every time. But you know, the positioning of the graph, it twists. It doesn't really matter. What matters is the connections that you have, the order of the connections, and the type of connections, which is pretty simple. Undirected here between the nodes. But of course, when it comes, it's a simple graph. So, you know, if I keep running and it changes the positions with a random positioning, you can still see it's like the same. But when you have complex graphs, which you want to visualize very cleanly, the layout algorithms, which layout algorithms, the network X has quite a lot of them. It matters what algorithm you're going to use. But for us, I don't know if we're going to get into visualization, but the key is to learn, like you'll see in the next lectures when we start like parsing out network data, real network data, real bioinformatics data from files, how to utilize those methods so we can create a graph with a real data set, right? And then the visualization, yeah, we can try different layouts to see if we can get a nice visualization. And I don't see any questions on the chat. Please ask if you have. I'm going to keep going forward in the code. Okay, so some more. Let me copy it over. I'm going to start a new cell. Okay, so here's my code. Okay, so here I added some comments. Comments you can always put with the hash sign. Oops. Here you can put it here, or you can put it on the top, right? Doesn't matter. Okay, so here you see how we added another node, but as I mentioned earlier in the slides, those nodes, we can add attributes either to nodes or edges, right? And this node I'm adding here, it has the attribute that it's a protein. Okay, so what is now with the attributes, the way the network X operates? And the attributes, like I said before, it's the data structure is generic, and it's up to you how you model the data, right? So this label that you see here, that's not something part of the network X. That's something I define, and I say this node is a molecule. And what type of molecule? The type of molecule is a protein, right? So basically, it's an attribute that I set up there, and then I give it a value, right? I mess it up. Being a protein. I mean, you could call it anything else, right? Type of molecule, or you could call it like, of course, it has to be like continuous, right? You cannot write like 3 words. Or you could call it like, entity equals protein or something. That's totally up to you. But then you have, once you select one, you have to be consistent throughout, right? So then I'm going to go and add a bunch of additional nodes from an array that you saw before, how we can add many nodes. And then those nodes, using the previously defined attribute which I selected to call it molecule, I'm going to give it a value of RNA, right? So what you see now is that I'm started building a little more rich data structures to represent the real world data sets. So I added these 4 nodes, one being a protein, one being the other 3 being RNA, right? And you can do all combinations of those, right? You could have like 2 proteins and 2 RNAs, whatever, right? Now, I added those nodes, but I still haven't added edges between them. So basically, if I was to draw the graph now, you would see like what you see before, and then you would see like a circle, a ball, a node, however you want to call it, number 6 just hanging here by itself, and 7, 8, 9 like floating there in space without any links, right? Because we haven't added the edges for them. And I guess I can put this a little earlier. So that's how I can add the edges, how you saw before, right? All together in one swoop. So here's the, say, add edges from. Here's an array of tuples. First tuple, second tuple, third tuple, fourth tuple, each tuple representing the links between 5 and 6, 5 and 7. So I'm connecting the 6 and 7 nodes to the 5 that I had before. So the 6 and 7 is going to be up here connected to the 5, and so on and so forth for the other ones. Okay, so now if I decided, oh, I would like also, I remember that my node 1, that was also a protein, and I wanted to update that, right? So that's what I'm doing here. I'm modifying attributes of existing vertex or node, however you want to call it. So basically, I'm going to my graph. I'm getting the nodes of my graph. I'm getting node number 1. And then for that node, I'm adding an attribute. And it has to be consistent, right? Because we're building data structure. So, we selected attribute molecule to represent with a string here, whether it's a protein or RNA, or we could call it DNA if we want it. That's fine. We're just making up like random stuff now. So we'll go back to our node 1, which we defined previously in the code, and we now add that attribute after the fact. That this node 1 has now also the attribute molecule, and we assign that the string DNA, right? So you can add the attributes when you define the nodes, right? One by one or all together. And then you have to link the nodes to the graph. And then for nodes you have defined already, you can go and add attributes if you want. That's no problem at all. Okay, so let me actually finish the code, and then we can do questions. All right, so now we want to draw this graph with those attributes. So that's where it's a little tricky to remember, but you see the code here. It's in the documentation of the library, so it shouldn't be any problem. What you need to get is to just say that the labels, because essentially what we want to do is like draw the graph, and it's of our circles have a label which says, based on the attribute molecule assigned, says it's a protein or an RNA or a DNA, right? So you just need to get those labels through these get node attributes. So NX, our network X library, the shortcut, when we do the import network X. So get the node attributes of graph G for the attribute we defined being molecule. And I used single quotes here on purpose, but you can use double quotes. I just use single quotes on purpose just to show you that you can do it both ways, right? And if you're wondering like why doesn't it have quotes here, why does it have quotes here? Well, I want to talk about the underlying foundation of things. This is a literal here, right? When you pass that to that method of adding node, and this here actually is a dictionary key, because the network X underline uses dictionaries. I guess it uses has like a couple of levels. It uses the NumPy, I think, or the SciPy, one of those libraries, which under that NumPy or SumPy is dictionaries, right? The very base data structure of Python. So it's like network X, NumPy, and then underneath NumPy arrays, which are actually based on dictionaries, right? But for you, it doesn't matter. That's the magic of object-oriented programming, that you just read the library documentation and focus on the high-level stuff, right? So going back to our visualization, if we want to add the labels on the nodes while we draw the graph, so we need to get the attributes we defined for that graph. So essentially the values for this attribute molecule we define, and store that in the variable labels, and then actually go ahead and draw that graph and say like NX draw, like we said before. G is our graph, and as labels use this data that's in this label variable, which, as you see, actually the Google is nice to tell us it's a dictionary, which we loaded these labels from the values of our attribute molecule, and then use our font weight bold. So let's go ahead and draw it. And here's our graph now. Of course, if I keep redrawing it, you get a different layout, like we said before, because you don't specify the layout, and then it just picks up a random one. But you see now the labels are appearing here. Those nodes, we haven't added anything. I guess now you don't see the node numbers because we substitute the labels with this attribute, but those ones are like which ones haven't we added attributes? I guess the 3, 4, whatever those, and this is the 1 which we added DNA. I believe this is the 6 which we added protein, and then this is the 7, 8, 8, 9, which we added RNA, right? So this is like the 3, 4, 5 here, I guess. Yeah, so this is our graph. And then you can keep redrawing and getting different shapes. Okay, so let's see now. Any questions in regards to those codes? And I can go back if you want. No questions. Okay, so either everybody understood everything or everybody understood nothing. So that's how the logic goes. But I'm sure like it's the first one. You guys understood it. So it's simple code. So okay, and again, refer to the full documentation of Network X, definitely the tutorial you're required to read, and the reference referring to it to solve the homeworks and understand the code. And if you're more inclined to read the whole thing, it cannot hurt. But definitely the tutorial is required reading. All right, so now going to the graphs for more complex bioinformatics data, a picture from the European Bioinformatics Institute tutorial, which also you're required to read. Undirected graphs, which is what we have been seeing now. Directed graphs, which we discussed, directed edges. So, this gene B regulates gene D. So it's a one-way direction. Gene C regulates gene D, and gene D regulates gene E. Or it can be a metabolic network, which is another thing you could represent as a graph. So, metabolite C gives metabolite G, and metabolite D, and metabolite D converts to metabolite E, and so on and so forth. Directed edges, that's how this data is represented. Now weighted the graphs with thickness of, I guess, thickness in visualization. It's more like the numbers in the data structure denoting the strength of that interaction. And then, when you draw it, you can use as option, which we'll have that in the homework, as an option from the Network X to draw like thicker edge, the higher the number it is, right? I mean, the visualization is a way to represent the data. The key is like what's in the data, right? And what's in the data is like bigger numbers represent stronger interactions, right? And this is weighted, undirected, because we don't see arrows, weighted, undirected interactions. But of course, you could have also weighted, directed interactions. And you'll see that if you read the Network X documentation, it shows you there are different types of graphs you can make: undirected, directed, weighted, directed, unweighted, directed, and so on and so forth. So, okay, and really quickly, an excursion to linear algebra, which will go into, I mean, that's not linear algebra, it's more like adjacency matrices for representing the network data. But if you want to think underneath what's going under the surface, so basically, how is this represented? Essentially, you put 1 wherever we have an interaction. So the B and the A, right, interact here. So you put 1, and the B and the A interact here. So you put 1. You don't have self loops between the nodes, so everything on their diagonal. So A doesn't have a self link with itself. Neither B, neither C has a self link with itself. So everything is 0 at diagonal. And here, because it's undirected, so whether you look at A interacting with B, right? A interacting with B, you put 1 here, or B interacting with A, you put 1 here. So that's adjacency matrix internal representation for undirected graph. And what the degree means is essentially how many interactions each one has. And it's very easy to get the degree. For example, he just puts it here on the outside of the matrix, but for A, the degree is 5, because A, I mean, you can see it here, 1, 2, 3, 4, and 5, the F, right? That's the degree of interaction with A, but you can also see it from here, right? You get the A and count how many ones it has. So 5 interactions, right? And for B, you see interacts with A, interacts with F, so 2 interactions, right? For B, the degree is 2, but you could also go for B and get the column here. You can get the row too, because rows and columns, it doesn't matter in the adjacency matrix of undirected graph, and see add up the ones, whether you go by column or by row, and see it has a degree of 2, right? Also, one thing here is like, you see how big this matrix gets for this little graph, and you can imagine the bigger the graph, the bigger the matrix. So, and a lot of it is zeros, so that's what we call a sparse matrix, and a lot of wasted space. So there's like some tricks there, which we'll see when I do the spectral, that internally the network X and other graph libraries used to kind of compress that and remove the wasted space by the zeros, right? Now the directed graph that we've seen before, again, we don't have self loops here, so A doesn't have a self-control loop. When I say self loop, I mean A regulating, auto-regulation, regulating itself, so that we have everything again 0 in the diagonal. But here A interacts with B, so A interacts with B, but B does not have, it has an interaction with A, but there's not a directed interaction from B to A, right? So this is 0 here, right? So we go from A to B. That's what is represented here, but not from B to A. So where it was symmetric, if you will, before in the undirected graph, here on the directed graph is not symmetric, right? This is like an educational kind of example where the graph is kind of like the directed flows forward. That's why all the direct interactions is in the upper diagonal of this matrix, right? And similar, you can see, like, for example, the C interacts with D, right? The C interacts with D here. Okay, and then the degree, the same way, you just add up the ones here, only along the rows, as this directed matrix is structured. And then now we have the weighted graph, where here is very similar, like how we had it before, but instead of so it's undirected, so it's symmetric, right? Interactions A to B and B to A, but instead of having just ones, there we put the weight of the interaction, right? So you see between A and B, the weight of interaction is 8. So A and B would put 8, and B and A would put 8 again, right? It's undirected symmetric. If it was directed, we would just remove one of those. And then the degree is essentially, I think this one, I don't agree. Again, I got this from the EBI. I don't agree how they have it. It's all what you want to represent, because the degree usually represents the number of interactions you have, and then that's not the number of interactions by what's the definition of the degree. That's the weight of the edge. So that's could be called cumulative weight. And for me, the degree is like how many interactions A has, right? With E, with D, with C, with B, and F. So it's like 1, 2, 3, 4, 5. So the number of interactions would be like, just count how many entries you have. 1, 2, 3, 4, 5. I guess why is it has 6 too? That's weird, because the A doesn't interact with G. Okay, so that's maybe another weird definition, because maybe it counts the weights of the path, but that's a different story. Let's not count that. So just ignore the G here for now. So what I would say is, like, here we just add up the numbers, and that's like the total weight of interactions, and the degree would be how many entries you have. 1, 2, 3, 4, 5. Please ignore the G. So it would be like degree 5 and weight of interactions, adding up those numbers, right? But again, that's those things wouldn't need to worry about. I just wanted to show the underlying data structures. These concepts will come later on when we discuss, for example, the spectral. But as far as defining our graphs, you see we do it very abstracted with a network X. And let me go back and see these questions. Yeah, so one question is A to G weighted 12. That's what I said as well. I don't know, that's how they put it. It shouldn't be like that because it's not connected. And another question is, what is a biological example for having different weighted edges? Essentially, there will be, there are many examples. One I could give is the strength of the interaction, right? How, if you have, say, a protein regulating a gene, how strong is that regulation, right? And when I say like how strong it is, then that's something that would be in the experimental data, because essentially in the bioinformatics research, what you would do, and you will see that in that class, when we get data from the databases, it would be they measured some using the yeast-2 hybrid protein interactions, right? And there's ways when you do these protein measurements to measure like how strong the protein is associated, right? And of course, this comes from like lab experiments, and in those lab experiments, when they do the measurements with the instruments, you get some numbers out which says this protein with that protein, they interact like, there's always like a normalization, and then you just get some numbers, and then the strength of the interaction is 10, and then this other protein with the other protein, the strength of the interaction is 15. So, you would parse out those data, and then the 10 and the 15, you would put the corresponding weight in the interactions as you saw here on that graph, right? So that's one example in how the data would come. Okay, so now let's go see the a directed graph, and you see we're using the network X directed graph. So let me get the code and open a new cell and just put it in here. Okay, so create a new graph, G1, which is a network X directed graph. And then the usual thing we do, we add edges from 1 to 2, 2 to 3, and so on and so forth. Okay, so one trick, which actually works before too, when you have undirected graph, you can try it, is that when you use the add edges method, so you see here, I created the graph, but I haven't defined any nodes, right? So I'm just going straight up and saying like add edges, 1 to 2, 2 to 3, 3 to 4. So immediately it's going to create the nodes 1, 2, and add an edge between them. And then next create the nodes 2 to 3, and add an edge between them. So you can save all that type into add the nodes and then add the edges, and so on and so forth, right? So that's another way to do it more efficiently. Of course, when we have real data that I mentioned before from experimental data, we're not going to be doing, I mean, we'll probably do it manually in the first, maybe in the first homework, yeah. But the whole idea is that you'll be reading a file which has, hundreds of lines with the nodes specified in their attributes, and you're going to have to write some code that you parse out. I'll give you examples that you parse out their file, that file, and as you parse it out, you just building the network and adding those edges, right? So that's in a real world scenario with real data, and we'll say that. But then here that we're just learning how to use this, and we're building our kind of toy graphs. I told you that you need to go and specify the nodes separately, just define the edges, and at the same time add the nodes as well. Now, if you want to specify the node attributes. You always have to do that. By going back to the nodes which we just defined. So as we've seen before, get from graph 1, node 1, and then add an attribute called name. You see, before I was using molecule, now I decided to use name in that graph. It doesn't matter what you choose. You could call it mol, M-O-L, for example. That's fine, as long as you're consistent, right? So now I chose name, and then I call it GName. And then I go for the node 2 that I added up here, and then this one, I'm going to call it GNB. So, if I was to parse a real world data set, which would have, like, say, would be the simplest case, like a 3-column file, first column being the first node, second column being the second interacting node, and third column being the name. I would just parse out that file and add the nodes and add their names as I go with a loop there, which that's where your Python coding skills come in. And yeah, so for the first 4 nodes, I manually added here gene A, gene B, and protein A, and gene C. And now, again, remember, this is a directed graph. I added on the fly the nodes and the edges. I added attributes being names for those nodes. And then I'm going to get, if I want to draw, as we've seen before, right, the node attributes, essentially the labels. So I'm going to get labels, get node attributes for G1 and name, right? Before we called it molecule, so we get node attributes for the graph G that we had before. We got the molecule, right? So this and this, paste it from, this would be under there. Okay, so after we got it, then we're going to draw the G1, labels as labels, and then just use font weight is bold. And there you are. Now we have the directed graph for node 1. And you see here that the nodes here, the integers, was used more like a placeholders, right? What really gave the richness to this data structure is when these integers, you can imagine like when you have an array, right? A plain array where you have the position 0, 1, 2, 3. I mean, I could have started from 0 here to, it doesn't matter. It's just integers as far as Python is concerned. But you could imagine like being an array with 0, 1, 2, 3 as placeholders for the nodes of the graph. But of course, more richer than the array, here we have the structures, the connections, directed connections between those nodes. The array is just like an order collection of items. Here we have some more structure which shows like where each node is and how it connects directed or undirected with each other, right? But then on those placeholders, we added the attributes, which is what we're interested in, right? And then we got those attributes as labels, and then we draw those graphs with those attributes as labels. And you see here, we kind of start seeing like a more real data structure for modeling biological problem, which is gene A regulating gene B, and gene B regulating protein A, and protein A regulating gene C. Again, that's kind of like, and something else gets regulated here, which I guess is the 5, because I didn't add the label. And it's pretty simple downstream regulation, and protein A regulates 2 genes also downstream, right? And there's many things you can do here, make a richer graph to sort of have upstream regulation too, like an edge pointing back, and of course play with the visualizations, which you can see in Network X, like how you can make a little more colorful edges, or thicker if you have weighted edges, and so on and so forth, right? So this is our directed graph. And going back to the chat, I see question molecule instead of name. We explained that it's an arbitrary attribute that we use. You could call it MOL. You could call it hello if you wanted. It wouldn't be very straightforward to understand what you store in this attribute, but just try to pick a name which is um, more self-explanatory. What is this attribute? Of course, we'll see that too. You can add like as many attributes as you want, right? You could add molecular weight. You could add like whatever else. Essentially, like I said, when you parse out like a file with bioinformatics data and you created graph data structure from that, it's up to you to decide like how to code that stuff, what kind of attributes to use, so that besides the the aspect of you later on or somebody else understanding the code based on the names you use in there. That's why I'm saying use something that is self-explanatory. It's also there can be bugs, code bugs, as you build the data structure, because you need to build it as correctly as possible to model the real world data, right? So that's a big part of the development there of those algorithms and those codes. Okay, so if there's no other questions for that, let's keep moving. I guess, yeah, we discussed that directed graphs represent direct control, for example, biological regulation. And as you saw in that graph, gene A, what would you represent here? The gene A regulates gene B, and gene B regulates protein A regulates gene C, and whatever that is here, right? So, okay, so that's with directed graphs. Now some more examples with the this tutorial, which you're required to read. You can do that stuff in Network X. We're just not going to show it here. You can also represent the size of the nodes to represent degree of connections, right? So this one, which has 5 connections, is it's a bigger node compared to like those ones that have a single connection. This one has 2, a little bigger than the one, and this one has 3 connections, right? And then we saw the degree also in the previous slide with the adjacency matrices. And then we say here, what you see basically that you visualize it in a way that the degree of each node is indicated and reflected in its size and color. And again, the visualizations is just something to convey what's underlying in the data, right? Like the degree, as you saw before, is on the adjacency matrix. Like in the network X, you're never going to see the adjacency matrix, but you're working a higher level than that, right? You let the framework like take care of what's under the underlying data structures for the raw numbers, right? But this anyway, the degree is inside your data, and then how you choose to visualize it, that's just for conveying like the message, if you will, right? It would be more important when you run like graph algorithms for the degrees and all that stuff to be captured correctly in the data, so the algorithms can give you the right answers. And just one, for example, question that you could answer with this algorithm, which is the most highly connected, one question would be, which is the most highly connected nodes in your graph, right? Your hubs, right? So you run the algorithms and it scans the data to find the nodes with the highest connections, right? And that's a very simple thing to do, but there's other stuff too, which is also like the paths, how central are those nodes, what we call as hubs in the graph. But I'm just going ahead of myself. Okay, so directed network nodes have 2 values, out degree and in degree. I guess I don't visualize that here, but essentially this one would have like, I guess we could see it in my graph too, but I chose that one. So this one has an in degree 1, and this one has also an out degree. So one coming in, 1 coming out. In degree 1, in degree out. So the D, for example, has an in degree 2 and out degree 1. So it's 2 coming in, and 1 coming out, right? The B has an in degree 1, out degree of 4. That's another thing to define. And then I guess here it just counts the degree here. We didn't mention before. It just counts the out degree, right? And how would you find the in degree? You would look at the column. So you see here, that's one useful thing from the adjacency matrix. If you look at the rows, you see the out degree of B, because the rows basically, it designates this directed graph, so it designates the directed connections from B to C, from B to D, from B to E, so that's from B to those, so that's the out degree, which is 4, where the columns here, it would indicate like what comes into B, right? Or if you want to see for D, for example, what comes into D, from B to D and from C to D. So there's 2 coming into D, so the in degree of D would be 2 ones here. So the in degree of D would be like 2 of them, 1 coming from here, 1 coming from here. But then if you go for the out degree of D, you see you get the row. So you see the D only connects with E, right, which is here. So the out degree of D is only 1, right? That's, not anything that applies to the stuff that we're doing, but since it was mentioned here, it's good to do it. All right, so some other graph algorithms that we mentioned. Let me go check questions. No questions. Okay, so paths between the nodes, essentially how many hops is to get from A to B, not counting the nodes themselves. It's like 4 hops, right? That's a graph traversal algorithm, graph path algorithm, right, that you can do. And essentially it's used to model traditional computer science, like how information flows, and like when you have graphs representing social networks, for example, or like even the internet, like regular data networks. But in our case, biology would be, again, in the same sense, how information flows. Like, for example, if we represent a cell signaling pathway, interacting proteins cascading a signal from the outer of the cell to the inner of the cell to the nucleus. So that's information flow too. But also you could say this is to model how distant proteins and genes interact. So A and B, they're not directly interacting with each other, but then this network traversal graph path algorithm to find the shortest path between the A and the B would be saying that to get from the A to the B, we have to get through this 4 interactions. So I guess it's a little more complex with biology because, it's not like those represent like cellular entities, which are not like balls and sticks, right? Like, for example, yeah, the algorithm might say like we have 4 steps, whatever this protein is degraded in the cell, right? I mean, in computer science, pure computer science, when you were like measuring, say, data transfer in the network, you could say, yeah, what if this server on the internet is down, so data doesn't flow through it? So it's kind of the same thing here, but in biology, things a little more fuzzy, right? What if this protein, like, is degraded, or it hasn't been produced in enough quantity in the cell, so it can, transmit the information and the regulation path in the network, right? But anyway, I'm just mentioning it because it's interesting, but I'm not going to go into like that deep detail. We'll just focus on the data, right? And algorithms calculate shortest paths on the graph, right? One example of those algorithms, and then if you again read on the Network X documentation, you'll see where it mentions some of those algorithms that it has built in. Okay, and then some other examples: scale-free networks. Scale-free meaning that it has those. That's actually these type of networks. We find quite a lot. in biological cells means that the central hubs, which make the network scale-free in the sense like even if the network is scales up, if the network like grows, because we have those hubs, we can go from any node to any other node in like a maximum of like 4 steps, right? 1, 2, 3 steps to get here, or 1, 2, 3, 4 steps to get here, right? Or from here to down here, we can get in 1, 2, 3, 4, maximum 4 steps. And there's some algorithms to calculate what's the maximum steps you can get from any node to any other node. But the definition of scale-free networks is that you have those central hubs connecting distant parts of the network so that you can go from any part of the network to any other part of the network to regulate any gene to any other gene, if you want to call in biological terms, in a maximum number of steps, right? But also, if you read up on the theory behind those scale-free networks, on that wiki article, I think it should be in there, which I had about the graph theory, you'll see these also, the central hubs create a problem because if those hubs break, and we have that in the cells, like P53, a central protein in the cells, it gets mutated, then the whole network breaks down because the hub, yes, it facilitates very fast flow of information and regulation, but also it's very central. So if it breaks down, the whole network can break down and can get disconnected. So that's another thing you have to keep in mind. Okay, and yeah, some other definitions: centrality, how central is this node, and how it facilitates the transition fast in least number of steps from one area of the network to the other, called the transitivity. And I have a link here if you want to read more details in regards to that. But this is pure network theory, which we'll touch upon a little, but our approach is going to be more from the data modeling perspective, and also like how to apply machine learning algorithms in those data to discover patterns, right? We're not going to do a lot of network theory, and I think that's it that I have for today. For the slides, we keep it, as I said, a little shorter. I always try to finish it between 12:15 and 12:30, so we have time for questions afterwards. So yeah, so that's it. Let's see, questions. Alternative hopes or paths can lead to path least resistance, may be important for diseases. That's correct. Fast signal transduction, change in cell responses to environmental stimuli. Yeah, all that goes under the same concept. Okay. Thank you as well. So I can stay a little more, like 5 minutes. Any questions you guys have regarding anything we discussed for the lecture material, on the slides, for the operation of the class, the Google Colaboratory, or any other items? And then the question is where the Zoom recordings will be posted. It will be on the Blackboard. So if it's actually recording now, then it's going to process through it, and then it sends me an email when it's ready with a link, and then I get that link and post on the Blackboard. So it's going to be there later today or tomorrow. I can stop the recording now.